work, and Walter Funk, the guy in the purple shirt that I showed you earlier, did some experiment where he took young and old human cells and he put the telomerase gene into the old cells, and he also had some old cells that he didn't put the telomerase gene into. And then he cultured these cells into skin on the back of a mouse. Now, you couldn't do this with a human because the FDA would never allow it, but you can do this with mice. So he looked at the young skin, the skin from the young cells growing on the back of the mouse, and they looked young. When he looked at the cells from the skin from the old cells, the skin looked old. But when he looked at the telomerized cells, the old cells that were telomerized, these cells looked young. Well, that was really great, but hold on. You can buy a lot of things in cosmetic stores that'll make your skin look young. So that's nothing new. So Walter took this one step further. He then did DNA array analysis. And DNA array analysis is a procedure where you can look at all the different genes inside of a cell and ask what their expression level is. So he took the cells from the young skin and he looked at it by DNA array. And this picture shows like a computer chip. Each square represents a different gene, and the color represents the expression level of that particular gene. So you can see a profile. This is what's called a young expression profile. When you look at the old skin, you see a very, very different expression profile because a lot of genes are reduced in expression, a lot of genes have increased in expression. But when we looked at the telomerized cells, he saw almost exactly the same expression pattern as the young cells. So for the first time on this planet, somebody had actually made cells younger. And again, I'm talking about cellular aging. So this was very exciting news. It said there was maybe a lot more promise in telomere biology than we originally thought. I'd like to take this one step further. We just looked at skin cells grown in a petri dish, then we looked at skin cells grown on the back of a mouse. Now I want to talk about a whole organism. Now mice don't age like humans do. They don't have telomere shortening. They have telomerase turned on in all their cells. And so it's a really awful animal for using as an animal model when we want to make drugs when we're trying to make drugs that turn on telomerase or prevent telomere shortening. But we were able to engineer, and when I say we, I mean Maria Blasco in Spain, was able to engineer mouse embryonic stem cells so that she destroyed the telomerase gene in those cells, and then she grew those cells into mice by cloning, and then those mice at first acted perfectly normal because I said mice don't age by telomere shortening. Their shortest stick of dynamite appears to be free radicals, but they're also very driven. Their death is by cancer in a majority of the cases in predators in the wild. So these mice were developed after the first generation. They still look normal, but the telomeres got shorter and shorter. Now the babies are born with shorter telomeres than the parents. So eventually, after 7 generations, the mouse telomeres got short enough so that these mice started aging just like humans do. Then she and you see in the bottom corner two other publications took these mice, crossed them into such a way that half the babies would have telomerase, half the babies wouldn't. These mice are the same age, they're from the same litter. The top mouse has short telomeres, the bottom one has long telomeres, and this top mouse is suffering from a lot of age-related illnesses that humans do. Gray and thinning hair, weakened immune system, intestinal atrophy, reduced spleen size, decreased wound healing, a very easy thing to measure in aging, and decreased lifespan. The top mouse lives on the average 2 years, the bottom mouse lives on the average 3 years. Okay, so that was the proofs of principle showing that telomere shortening does cause replicative senescence and cellular aging. And so now in answering this question about why do the green cells appear not to show any cellular aging and the orange ones do, we can say that telomerase, the green cells express telomerase, and that's a typical example of that is the human embryonic stem cell, which expresses a lot of telomerase and has no telomere shortening. And the somatic cells don't have any telomerase. Okay, again, why? Well, even though those cells, the somatic cells, didn't express telomerase, they still have the gene for telomerase. And the gene in the germline cells is producing telomerase. Remember the gray factory thing that I showed you before? This is showing the gene producing the telomerase protein. Now, in the case of a somatic cell, that repressor is, there's a repressor protein that binds to a repressor site, in theory, and shuts off this gene. And so what my company and Geron Corporation spent a lot of time doing is we looked for an anti-repressor that would bind to that repressor protein, thus preventing the repressor protein from binding the repressor site, thus turning the telomerase gene on and lengthening our telomeres. Well, the very first chemical that was ever discovered that would actually do this just came a few years ago, and it's called TA65. And this is a nutraceutical, and it's distributed by TA Sciences, and it's available for research. And they've provided my company with a lot of this nutraceutical so that we could do some research on it and study it. Remember, I'm a research scientist. And so some of the experiments that we wanted to do was, one, validate, does TA65 really turn on telomerase? And we want to do a few other things. Well, to validate, to measure whether or not telomerase activity exists in a cell, we did a procedure called the TRAP assay. And to explain that, I just want to go through what a telomerase assay is, how it works. Okay, so you take a DNA, a synthetic telomere, and that's what this green thing is here. Telomeres are made up of a repeated unit TTA, GGG, TTA, GGG. And this just has that repeat in there 3 times. If you mix this synthetic telomere with an extract of cells that contains telomerase, such as, hopefully, cells treated with TA65, telomerase will bind, add 6 bases, pause, move over, add 6 bases, pause, move over, add 6 bases. And that's how telomerase works. And it's the only enzyme on the planet that works that way. So hopefully all of you are familiar with electrophoresis, and I won't have to explain this, but if you take those synthetic telomeres, the lengthened one, you'll find that they actually form a 6-base ladder. And if you see that darkest 6-base ladder, that's actually a positive control that we use in our experiments. It's a cancer cell called HeLa, and HeLa constitutively expresses telomerase. So we can see that telomerase activity by seeing that ladder. Now, the next lane over is just showing the same cells, but using 1/20th as many cells, so the ladder is a little fainter. But now the next 2 lanes shows a skin fibroblast, a human skin fibroblast cell line that's not cancer, that's very normal, and it's just as many cells were put into that lane as the 100% HeLa, and there we see no ladder at all, and that's because these cells don't express telomerase. Okay, so the goal of Geron Corporation and my company was to find some way of turning those cells into cells that would produce the ladder, but at the same time not being cancer. And so we've been for years testing everything that came along. Every time somebody published saying that they have some idea how to turn on telomerase, we would test it. Always negative, always negative. Every time somebody would call us up saying they found something that turns on telomerase, we tried it. It was always negative. Try TA65, and lo and behold, we see ladders. Now, I'm going to use a pointer now. You can see the ladders are faint, but they're very visible. They're faint and partially visible, but they are really there. So TA65 is the very first time we've ever seen something that actually induced telomerase expression. And as you can see in the lanes that don't get the drug, there is no ladder at all. Well, it was faint enough that we thought, well, maybe this is just some artifact, maybe it just got some weird thing happening that day. So we repeated the experiment, and again we see it. Now in this case, we can see a little more dose dependency. The higher doses are showing more of a ladder than the lower doses, which is good. So we repeated it again, and again we get this. So we repeated this several times, and we very reproducibly can get TA65 to turn on telomerase in normal cells. And it's the first nutraceutical, chemical, anything on the planet that can do this. As if there was something, well, let's say there's a few other ones now, but I'll talk about those later. TA65 is also shown not to be toxic, at least by the experiments that we've done at Sierra Sciences. One is to just, we test a lot of drugs at Sierra Sciences, and we always see toxicity to some degree or another. And this particular nutraceutical was showing practically no toxicity whatsoever, even at the highest doses. So if you look on the right-hand side of the slide, you'll see cells treated with nothing except DMSO, and then the other cells treated with TA65. And I don't know how clear you can see it, but you can see that the cells look exactly the same and about the same density. We did a lot of other tests for toxicity, including just cell counts in a culture counter, cell titer glow, which is measuring ATP levels, and PsyQuant, which is measuring nucleic acids in the cell. And in all cases, we saw no or very low toxicity. Another thing that we did to kind of look at toxicity is we did DNA array. Remember I talked about DNA array before in that human skin grown on the back of a mouse model. In this case, we took cells treated with TA65 and then asked what happened to the expression pattern. So we had cells that were untreated and then cells treated with TA65 and asked what kind of differences did we see. This graph is a little hard to explain without a pointer, but let me try it. The x-axis is just showing the full difference in expression. So if you see that number 32, that says 32 different genes showed a twofold decrease in expression levels. And then 9 genes showed a twofold increase in expression level. Well, that's practically no change at all, if anybody who's used to looking at DNA array results. And what I want to show is that Sierra Sciences is trying to develop drugs too, and we've actually now discovered about 120 drugs that turn on telomerase, and we're trying to develop those, but none of them work as well as TA65 yet. And this is our best case of doing DNA array analysis with our best drug, C005684. And if you were to analyze this data a lot, you would see that about 10% of all the genes in the entire genome are affected at least twofold in their expression level. So TA65 is about the most inert chemical that I've ever seen in a DNA array analysis study. So TA65 is a nutraceutical extracted from the traditional Chinese medicine astragalus root. TA Sciences did some studies themselves, and their first study was to test 36 men ages 60 to 85 in a 24-week study. So this is the results of the study. They looked at immune system improvement, vision improvement, skin improvement, sex improvement. Now, even though you can see there's a trend, but a trend that the blue bars are treated with TA65, the gray bars are not treated with people that didn't get TA65. And you can see that there's a trend that people who treated with TA65 were showing improvement in a lot of these tests here. Now, if you look closely, you'll see statistical scores aren't that great. There are statistical scores like p.12, p equals 0.47. So the sample size was too small. But look at this score here. This is the immune system, lymphocyte proliferation. There was an improvement in lymphocyte proliferation in patients taking TA65 that was significant to the 0.005 level. Now, a lot of people, especially me, believe that the number one cause of aging is a weakened immune system. And so it's good to see that TA65 is having such a dramatic effect on the immune system. Now, there's a lot more data on this. I'm not going to go through all of this, because especially since it was a small pilot study, but you can see the entire study at www.tasciences.com. Now, in the last 2 years, a lot of people have been taking TA65, and that includes, I'm one of the people that's taken TA65. I've been taking the drug myself for 2 years. And a lot of these people that have been on for more than a year, finally we're offering some data. We've been getting our blood samples taken like every 3 to 6 months, tons of biomarker studies. You had to agree to all that in order to be able to purchase TA65. And after looking at a lot of the clients that have been on it for 1 year, definitely seeing some good things happening here. CD28 negative T cells. And I'm not exactly certain what CD28 negative T cells are, except they're T cells, and they've been heavily published in the literature to be very correlated with aging. When you age, the older you get, the more CD28 28 negative cells you get.